Good laboratory practice (GLP) is a quality system that governs how laboratory activities are planned, performed, monitored, and documented. In microbiology laboratories, GLP is crucial to ensure valid results, biosafety, and effective contamination control. GLP forms the foundation of a reliable microbiology lab by promoting consistency, accountability, and high professional standards. With proper implementation of GLP, laboratory work can be carried out safely and systematically while maintaining the integrity and quality of scientific results.
Proper laboratory organization and workflow are essential part of GLP. Clean workspace reduces errors and prevent contamination. In microbe lab, different activities such as media preparation and culture handling must be performed in separate areas. Clear labeling of samples, reagents ensure traceability and avoid mix-up, helping produce accurate and reliable results.
Equipment handling under good laboratory practice. Proper handling of laboratory equipment is essential to ensure accurate results, prevent contamination, and maintain the safety of laboratory personnel. Under GLP, all equipment must only be used by trained personnel and according to established standard operating procedures or SOPs. Before any equipment is used, it must be inspected to confirm that it is clean, calibrated, and functioning properly. Any damage or malfunctioning equipment should be reported immediately and must not be used, as it can compromise both safety and the reliability of results. During laboratory work, equipment such as microscope, incubators, centrifuge, biosafety cabinets, and autoclave must be handled carefully to avoid spills, breakage, and unnecessary exposure to microorganisms. After use, all equipment must be properly cleaned and disinfected, then stored in their designated location. This step is important to prevent cross-contamination, maintain equipment condition, and ensure readiness for the net user.
Correct waste handling protects both staff and the environment. In the lab, we separate waste into 4 categories: biohazard, chemical, sharps, and general waste. Biohazard waste is treated in the autoclave before it is disposed of. Chemical waste is placed in clearly labeled containers and must never be mixed unless approved. Sharp are discarded only in puncture-proof containers to prevent injuries. If a spill occurs, we contain the area, apply disinfectant, wait for the correct contact time, wipe it safely, and report the incidents. Following these steps reduce risk and keeps the laboratory safe.
Proper documentation is essential in the laboratory. All activities must be clearly recorded, accurate, complete, and legible so that any deviation or incident can be traced and addressed. Record keeping covers samples, test results, equipment maintenance, staff training, and incidents. All records should be dated, signed, and securely stored, which helps with audits, troubleshooting, and quality control. Standard operating procedures, or SOP, provide clear and standard instruction for lab work. All staff must read, understand, and strictly follow SOP, which should be easily accessible and regularly updated to maintain safe and consistent laboratory practice.
In our laboratory, biosafety is a top priority. First, all personnel are properly trained and competent. They understand the risk of handling biological materials, strictly follow standard operating procedure, and wear appropriate personal protective equipment to ensure safety. Second, all biological specimens and microorganisms are handled safely using proper techniques. This prevents contamination, exposure, and laboratory-acquired infections, keeping both staff and samples protected. Finally, any incidents such as spills, accidents, or exposure are managed promptly according to SOPs. Proper reporting and corrective actions help prevent recurrence and maintain a safe working environment for everyone.
As conclusion, good laboratory practice GLP ensures safety, accuracy, and reliability. Proper GLP reduces errors, contamination, and risk in microbiology labs. Everyone in the laboratory is responsible for maintaining GLP standards. Following GLP helps produce reproducible and trustworthy results.